Thank you so much, everybody, for joining in today. It's great to have you with us for a really interesting ARI seminar today. I would like to acknowledge that I'm joining you from Wurundjeri country. I acknowledge the traditional owners, past, present, emerging leaders, and I acknowledge the traditional owners and First Nations people all around from where you are joining from today. It's a pretty cool day here on Wurundjeri country. But lovely to see rivers running and lots of things happening out on bush. Today we've got two speakers for you. Neither of them are really ARI staff, but they're definitely people with whom we have collaborated and worked with in many ways, and we're really grateful for them coming along today and presenting seminars for us, both on topics that are of intense interest to many people. First, we're going to hear from Graeme Coulson. Many of you will know Graeme. One of his hats is University of Melbourne, but he wears many, and he's going to speak with us about how we can use translocation to manage landlocked kangaroo populations. And then we're going to hear from Rees from CSIRO. He's going to talk about how we can use fox control and how that might influence feral cat populations. So two really interesting seminars about managing various populations, quite different contexts. I'm not going to give them long introductions. I think you probably know who these guys are, but look them up more if you need to. So thanks so much for joining in. We'll hold questions. We may have questions at the end of Graeme's, and then we'll certainly have more at the end. If you have questions, please pop them in the chat, and then we'll come to them either in the middle or at the end. And I invite you now to sit back, listen to Graeme. Thanks, Graeme. Over to you.
Thanks, Fern. As Fern said, I'm going to be talking about a trial translocation that we've been running over the last couple of years and evaluate translocation as an option for managing kangaroos. Before I get into the details, I want to acknowledge the research team. This has been very much a team effort. So those people on the list there have played key roles in this project. And we've also had support from a lot of individuals and organisations listed down the bottom there. So a little bit of background first on translocation. What is it? It is simply the deliberate moving of an individual animal from one place to another. And you'll be very familiar with what are called conservation translocations. That little eastern barred bandicoot down there has been translocated to French Island. So some of these conservation translocations are reintroductions back to the former range of the species. In the case of French Island, this comes under the heading of assisted colonisation, where we're moving animals to a new location, and French was chosen because it was a fox-free haven. Another category of translocation is what are called mitigation translocations. And a good example of this would be pesky polar bears in Inuit villages, causing all sorts of trouble and threatening human life. So for these sorts of translocations, we're talking about mitigating human-wildlife conflict and also impacts on animal welfare. And that's the sort of translocation we're talking about today. So kangaroos, we're talking grey kangaroos here, are often the victim of development where they end up in what we call landlocked populations. So these are patches of remnant habitat with small kangaroo populations remaining that become completely surrounded by development or other inhospitable habitat. And that leads to all sorts of concerns about the individuals themselves and impacts they may have on people. And the sort of options that are thrown around are culling, which is obviously not very popular, particularly when you have residences right next to the site. The construction or enhancement of corridors to allow kangaroos to move off the site into somewhere safer, but they are remarkably reluctant to move. And translocation comes up a lot. In other words, if they can't be culled, then they won't go by themselves, and they be picked up and taken somewhere safer. There's not a lot known about translocation of kangaroos, but there have been a couple of studies. And the first of these comes from the Gold Coast some years back, a place called Harbour Town. Very small sample. We just had 13 kangaroos. And I'll just run you through the basic outcomes of that. 23% mortality during capture. So 3 of the 13 died while they were being attempted to catch. Survival doesn't look good. So this is 100% survival at the top, down to 0. It's a bit misleading because round about this sort of time period, the radio collars that were on these animals began to fail. So they lost track of the kangaroos. So whether these are genuine deaths or simply where they disappeared off the radar, it's hard to interpret. And the other outcome is dispersal. 2 of them remained as for the first year of the study, or the full year of the study. 8 of them dispersed and up to 6 kilometres, but there was no sign of directional movement in that. They seemed to disperse in all sorts of directions. The second study that's been published is one from Western Australia. So these are Western greys, a much bigger sample, over 150 kangaroos, and very high mortality. During capture, 41% of these animals died during capture. And then survival after capture was pretty bad too. This is the flip side of survival. This is mortality rate. And there are 3 sets of figures: the collared animals in green, others that were tagged but not collared, and the brown ones are inferred from the rate of loss of the other 2. And it works out to 9% survival in 1 year. Or if you look at the flip side, 80% of them were dead within 2 weeks. Dispersal, there weren't a lot to disperse, obviously, from that very high mortality rate, but 2 remained on the site, were sedentary. 4 did disperse, and they went up to 28 kilometres with a hint of directional movement back towards the source site. So with that sort of background, DELP has a quite detailed translocation policy for eastern grey kangaroos, and it sets a number of minimum requirements, which are quite rigorous, covering these 6 things. So you have to find a suitable release site. You have to develop strategies to prevent the kangaroos from going home. It has to be very much anchored in good animal welfare. Also have to consider human safety. The transport requirements have a lot of detail around them, and there's a very strict requirement for monitoring after release. So we set up our project to effectively test the assumptions behind the DELP, now DECA policy. And to do that, we needed a number of approvals. So the University of Melbourne Animal Ethics Committee approved all of the animal aspects. DELP provided a research authorisation to conduct the work. Parks Victoria had an access agreement with us because we were releasing into a park, and the conservation regulator had compliance staff who were watching our every move, both at the capture end and the release end of the program. Our evaluation is based on these 3 things. We wanted to look at the capture phase, the transport phase, and the release phase, and we evaluated release in terms of survival of the kangaroos and their dispersal. So our source site was called Kinley. It's a new development right in the heart of Lilydale. It's centred on this old limestone quarry, which is now closed and is being rehabilitated. And the kangaroos themselves hung out down this edge down here, which had some remnant native vegetation. And they'd also come out and feed on these newly terraced areas where the first release of housing blocks was going in. But as you can see, it's pretty isolated. This is the Central Business District of Lilydale. This is the Maroondah Highway coming through here. This is the new Lilydale train line coming through here. We conducted a survey to see how many kangaroos we were dealing with. So in December 2022, we did a sweep count and got a total of 56 kangaroos. You can see all the surrounding development there, the other houses. And the release site, chosen after a lot of searching, was Warramate Hills. It's variously labelled as a Flora and Fauna Reserve or a Nature Conservation Reserve, managed by Parks Vic. It has two hills, Steel Hill and Brighty Hill, that are well above the surrounding landscape, which together give you a pretty much 360-degree radio view of the area. It's a fair size, over 500 hectares. It was 12 kilometres from Kinley by straight line, but about half an hour or more driving. Has quite a habitat, a diverse range of habitats. It has a resident population of kangaroos and has other herbivores like swamp wallabies, wombats and sambar. The particular bit that appealed to us was this area here, and there's another area of semi-cleared land here. So this is the specific release site. It's an area of revegetated farmland. You can see the replantings there and some old tree guards. It has abundant food resources, and the La Niña years of the last couple of years have meant that food has been maintained. Good cover surrounding it, and no threatened plants. We weren't posing a risk to the park as far as we could tell. There are some threatened species, but not in these sorts of habitats. So our procedure, we started out with capture. If you keep your eye on the figures up in the left-hand corner here, you'll see how the numbers ran. We captured 37 adult kangaroos, and the 10 in brackets indicates 10 sub-adults, juveniles, that we captured as well. This was done between May and August in 2021. We started out with just 4 captures in May and then were stopped by a lockdown, resumed in July and caught another 9, and that formed our pilot study. And then a report back to the ethics committee gave us approval to go on to the full stage. So in August we caught all the rest. It was done mostly from one of these 2 vehicles, but a little bit of work on foot. It was all done late in the day, generally twilight and through the night up until about midnight, using night vision gear, and that ran over 11 nights. When animals were caught, they were brought into a shed on site for a full examination. We did a number of standard measurements, body weight and length and all the rest of it, external examination for injuries and any other signs of ill health, a blood sample from each individual, and 2 were euthanised at this point because they had existing injuries that would have made them very vulnerable to mortality if they were translocated, so they were euthanised at that point. The marking was crucial to this study because it was necessary for monitoring. So each individual was given an ID number and a name, and we named them after suburbs of Melbourne. You'll see those names soon. A subdermal pit tag, more as a backup than anything else. A reflective ear tag combination, so left in the right and red in white in the left and red in the right, tells me that this is Fitzroy. And he's also got his number on the tag as another backup. A plastic collar, which was soft and flexible and designed to fall off for the 10 young because they're too small for a radio collar, and then a VHF collar for all the adults. The collar had pretty good performance specs, had an optimal range of 10 kilometres, but that's reduced quite a bit with hills and valleys and things, a battery life of 5 years, and a movement sensor that would tell us if it had stopped moving and was likely to be dead. Transport was in a van, or later a dual cab ute, all padded and extra covering with a wool pack, a heavy wool pack. We did have 1 death in transport. So we released 34 adults, 22 males and 12 females, and another 10 juveniles. They were put into secure separate locations so they wouldn't disturb each other. We monitored them from a distance so we wouldn't disturb them, and checked that they'd gone the next morning, which they'd all had. The monitoring was based largely on the radio tracking, and we started with quite intensive monitoring, doing it daily for the first 2 weeks. then twice weekly for the next 2 months, and since then twice monthly for the next 2 years, which is coming up in August. Survival, first of our results. There was quite a lot of death, and I've listed the causes down here. A lot of animals ended up in water, presumed drowned. 5 road kills, 4 shot, 2 tangled in fences, 2 that were euthanized when they were picked up elsewhere, and 2 where we simply don't know what happened to them. And you can see the survival graph here. 3 died within the first day or 2, and then another one shortly after. There was another pulse of deaths here, and then it continued out. When we get to the 1 year mark, that's 40% survival at that point for comparison with those other 2 studies. Now we're out to 38% survival, and there is also 3 shown with question marks here that we don't know what happened to them. They're simply missing. If we counted those as also dead, the survival would be poorer, but this graph shows that without those 3. Settlement, did they settle in? You'll see a few of these maps. Red is dead and green is alive. These 3 were the ones that disappeared died very early in the piece. They ended up in Woorayallock Creek within a day or 2 of release. So they clearly didn't have a chance to settle, but all the others did, but died of various causes that I've just outlined. So we now have 1, 2, 3, 4 alive at Warramate. Dispersal. There was a lot of dispersal. If you look down in the left-hand corner, you'll see Richmond. This Dan Murphy's outlet is right on the highway that I've forgotten the name of, and clearly not a safe place to be. And the interesting thing about dispersal, I think, is that it didn't just take place and then everybody settled. It continued on. So these are days after release. 4 months after release, we were still having individuals disperse away from Warramate. And some of these individuals continued to disperse for a number of months. They'd apparently settle and then they'd go on again. And these are the dispersal distances, and they are huge. So again, red is dead. These are all roadkill here, or 1 fence entanglement, another roadkill. We'll come up back to these. We've got 4 alive here, and some really big distances. So Clifton Hill was the furthest mover. He's moved 25 kilometres. And we know he didn't go straight there. He actually worked his way down the western side of the Dandenongs here and finished up in quite a nice spot, but then managed to get himself hit on Burwood Highway. And Clorinda down here is actually in the Cardinia catchment. And homing. This is not what you want if you're doing a translocation. This big billboard was on this corner here, and some of the kangaroos took it literally. Edith Vale managed to get back into the development area and later died. She was another drowning. Gembrook got as far as the boundary. She paced the boundary, couldn't get in, and then she'd come backwards and forwards until she was eventually hit on the road here. Richmond spent a lot of time up here, worked his way around the edge of Lilydale, came through here. That's where he was at Dan Murphy's, and the next day he was killed here. Watsonia up here, another case, she got as close as here, almost home, and then retreated and is now hanging out on the edge of Lilydale. So conclusions. First of all, unlike the other 2 studies, I think that this trial produced very clear results. There's a couple where we don't know the cause of death, and we have 3 missing animals, but they're a small proportion of the animals we monitored. The capture and the transport were very low risk. We had no death during capture and only 1 in transport. However, survival is poor, so it's depending on whether you count those missing ones as dead or alive. It was 41 to 47% at the end of the first year, and it's now down to 38%. Dispersal is common, so at least half of them moved in the first year. The dispersal distances are unprecedented. We've never had eastern grey females move anything like those distances, and the distance that Clifton Hill, the male, moved is a new record for its species too. And of course, homing occurs sometimes. So I'll leave it there, and thanks for listening.
Graeme, thank you very much. That is absolutely fascinating. Tess, I think we might jump to a couple of questions because, Graeme, there are a few in the chat that you can probably see.
Yep.
I might start off with a couple of questions, particularly around survival, because that clearly is a big issue here. And there's a couple, I might cluster three of them together. One is, so what's the natural survival rate of these sorts of roos without being translocated? Because, you know, what's normally happening? And those very early deaths, do you think that's because they're unfamiliar with the environment? So they're, you know, going places where they're a higher risk or encountering risks in different ways. And, you know, you mentioned drownings. It's really interesting, quite awful. There's so many drownings. Are dogs playing a part in that, or why are so many of them drowning? Or is it that unfamiliarity thing? So there's quite a few questions about the survival thing. Can you start us off with a bit of that? Thanks.
Right. Remind me about the other two. The first one was, is this abnormal? And it's actually a really difficult question to answer. If we'd had lots of time and resources, we would have done a parallel study on the local population to find out how it ticked, ideally well before the project started, but that wasn't realistic. And as it turned out, the local population would only be part of the story because the dispersing animals joined other populations in a much larger area. So we don't have data on the background level around that area. The best results come from the long-term study on Wilson's Promontory of marked animals, and they have annual survival of between 80 and 90% for adults. So this is way poorer than that. And in fact, if you're running a reintroduction for conservation purposes, this would be regarded as a failure. It's simply the mortality rate is too high, survival's too low.
Yeah, okay.
Second question?
The second one was about, you know, those early deaths. Is that because they're not familiar with that environment and are dogs playing a role in them ending up in creeks and drowning?
Yeah. The early deaths, the first three were almost certainly still recovering from the anaesthesia. Xylitol takes quite a while to get out of the system, and it coupled with unfamiliarity, we think what happened was that they went down a grassy track, saw the Yarra River in front of them and did a sudden right turn, and that put them into Woorolloc Creek, which has very steep sides and quite fast flowing, and then there's a lot of snags in there as well. Three of them ended up almost in the same spot. So it might have been a sort of a visual trick almost, but very much compounded by the fact that they weren't with it and they didn't really know where they were. There was a lot of sign of general unfamiliarity. We'd often see our kangaroos on the management tracks inside the reserve, whereas we'd never see local animals there. So they'd use the tracks, still trying to figure out where they were, and they'd turn up in some funny places, like on that first slide, they were out in the middle of a paddock in the middle of the day. Local kangaroos just didn't do that sort of thing. The water, I think, is partly, as I said, with those three, they bump up the figures for drownings, but the others were on small dams, and the one at Kinley itself was in a silt trap, a very deep water body there, but certainly shouldn't have been a lack of familiarity because she'd come from there in the first place. We suspect that dogs are involved. Kangaroos have a very strong instinct to go to water if they're being pursued by dogs. There were certainly farm dogs around Warramate, and a number of the neighbours said that their dogs would chase kangaroos. So that's probably accounting for the others.
Thank you, Graeme. You touched on the resident kangaroos, and there was a question. Tess, you might be able to help me. There was a question there about did you think that the released roos might adversely impact the locals, and was there some assessment of carrying capacity, etc.? What about those sorts of questions?
Yes, that's one of the concerns about a translocation. The policy covers those sort of questions. So we had to do an assessment of the local population density. We didn't have a good estimate, but we were encouraged by the fact that there was so much food, and that was partly to do with the La Nina conditions. So we didn't think there was any possibility of food competition. And as it turned out, most of the resident kangaroos and most of ours would spend a lot of time out on neighbouring vineyards. They'd be feeding in the grassy rows between the grapes anyway. So there's tons of food. We don't think that was a limiting factor.
There's a couple of questions suggesting that the location was suboptimal for a translocation. I'm guessing that you didn't have many options, but do you want to comment on was this the best place, basically?
Well, as I said, we searched high and low. We looked at a lot of possibilities. It's very hard to know really what is going to be a good spot and what is not. Partly, if you do choose somewhere that's got lots of kangaroos, then you've got that competition question. There were more disturbance there than we would have liked. There was illegal shooting on the north side of the reserve, but there's illegal shooting goes on all over the place. And the more we've tracked our animals and met the people who live in the places where our roos are dispersed to, the more we hear stories about what goes on out there. So yeah, I think it's difficult. You can do a lot of searching, and you've got to find somewhere that matches as best you can. But it's interesting that really nice looking bit of habitat that we chose for the initial release, there's only one female who lives there now. All the others have gone some distance or a lot further, as you saw from the dispersal map.
Wow, okay. We've got 2 minutes for you, Graeme, and I'm going to pick one of the trickier questions.
Yep.
Because a few people have also asked this question, Tess. You might want to add in here. So what's the most humane option here? What should we do?
Well, I think that depends on how you weigh these things up. If you are concerned solely about stress, potential suffering, then you would probably cull on site back at the source site. If you wanted to give the animals a chance, then you would perhaps consider a translocation, but you would have to recognise that will carry a lot of risks for the animals, and there's a strong likelihood they won't stay where you put them, and they will then expose them to a whole lot of other risks. Those ones that covered 20 or 25k, imagine the number of fences and roads they managed to cross safely before something finally knocked them off. So it's not an easy decision, but I think what we've done is provide the data that helps to weigh up those pros and cons.
It's not an easy one. Graeme, thank you so much. This is a really useful study, really useful information, and I can see that it's generated a lot of thought, commentary, pondering from others in the audience. When we switch over to Matt, if you wish, you don't have to do this, but if you wish, you're welcome to jump in and reply to some of these if you want to, if there are concise answers that you can provide. Some of them you probably need to sit down with someone for an hour and chat it through. But if there's something concise that you want to reply direct to some of these, feel free to jump in on the chat and do that. But I would just like to say thank you so much. That was just fascinating, sad, and a thing that we need to deal with, and really necessary question. So thank you so much. And I'm now going to jump over to Matt. Thank you, Matt. More gnarly questions, slightly different context here though, about whether fox control might influence feral cats. So over to you. Thank you so much.
Thanks, Fern. Alrighty, yeah. So I'm Matt. I'm coming at you from Brisbane at the moment on Turrbal and Yuggera country. So here I'm a postdoc at CSIRO, and I'm looking at new methods to estimate the distribution and abundance of some key invasive species across Australia. But I'm going to be talking about some of my PhD research I did at the University of Melbourne. Is my toolbar coming up? Oh, well, there we go. Yeah, so I did a PhD at the University of Melbourne. It was pretty fun. I did it supervised by Brendan Wintle and Bronwyn Haradsky, and also Alan Robley from the Arthur Ryler Institute. And I had a lot of help from the Conservation Ecology Centre, so Jack, Mark and Emma in particular. And this was a project that was largely funded and designed with Parks Victoria and DECA. Yeah, and it was all about looking at the outcomes of fox control. So outcomes on foxes themselves, as well as feral cats, and also on two threatened native prey species, the long-nosed potteroo and the southern brown bandicoot, who foxes and cats love to eat. So yeah, so cats and foxes are really largely blamed for a lot of our extinction of native mammals in Australia. It's so accordingly, we spend a lot of conservation resources on fox control, primarily through poison baiting, 1080 poison baiting. So 1080 affects, so cats and foxes, eutherian mammals, are much more susceptible to this 1080, whereas our native marsupials can handle much less susceptible to the 1080 poison, which is why we use it. But because foxes are scavengers, it's really effective for them, whereas cats would much rather live prey. So unless they're really food stressed, they don't go up and dig up these baits. And there's also some like legislative issues, which makes it harder to control feral cats. So while there's a bit of cage trapping, foxes are much more commonly controlled at broad, spatial and temporal scales, whereas cats aren't. And so when we do fox control, we see that for about half the time, native prey, really benefit from it, which is great. Particularly brush-tailed possums, they definitely love fox control. But in the other half of times, native prey might not respond to the fox control, and there's even some like pretty concerning stories of native prey declining following fox control. So this might be because we're simply not killing enough foxes to take off that pressure from a particular native prey species. Or that native prey species might be succumbing to other threats in these environments, such as a massive fire. But a really big concern is that fox control might actually be benefiting feral cats. So foxes and cats compete for many of the same resources. Foxes are a bit bigger, so it might be scaring feral cats. So where we take out the foxes, cats might have more free reign over their environment to do what they need to do. And also, because there's less foxes, there's more prey for cats as well. So this is what we often call we call this kind of mesopredator release. It's pretty controversial in Australia. And I think that's because there's just not much evidence, like the evidence for it is quite limited. There's not that many experimental designs which show this. And so these kind of questions, particularly this mesopredator release phenomenon, were really strong things I wanted to answer in my PhD. So I went out to Southwest Victoria. I had like two kind of study sites where I wanted to do some experiments and replicate it across these two different environments. So the first one is the Glenelg region, which is Guntage Mara country. And the second is the Otway Ranges, which is Eastern Mara country. And I spent a lot of time on Gadabanud country in the rainforesty section of the Otways. So I want to pay my respects to their elders. It was really amazing to learn about these landscapes and be out on these countries. So in the Glenelg region, there's a fox control program called Glenelg ARC, which has been controlling foxes through 1080 poison since 2005. Whereas in the Otway Ranges, there's a fox control program called Otway ARC, which has been controlling foxes more recently since 2017. And so so it's really amazing, I think, both of these, like having these broad scale management programs which monitor that management experimentally. It's quite rare, so I was really lucky to be involved with these. And so now these programs are monitored using camera traps, which are these white dots you see everywhere. So I collated monitoring data from the Glenelg ARC program, and the Otway ARC program. And I also chucked out 1,000 camera traps over 3 years. So all up, we had over 3,500 camera trap deployments at over 1,000 sites from 2013 to 2020, which I'll talk about today. And these landscapes, there's no dingoes here. Unfortunately, quolls have really disappeared from these landscapes. But it means that cats and foxes are like pretty much the only large predators in these environments that live on land. And so it gives us a really good focus on the interactions between these two species, like what's going on with them. So yeah, I stuck out 1,000 camera traps with a lot of help. So I think nearly 50 people helped me with my PhD research out in the field. Yeah, so we got to borrow camera traps when the weather was bad and no one else wanted them. So lots of walking to camera trap sites. And yeah, camera traps are really, really cool for just see some amazing things like interactions between different species, and yeah, really good at monitoring the whole kind of community of animals. That is, if you can kind of wade through millions of images of wallabies grooming themselves and carrying their little piles of debris, and kangaroos just napping for hours in front of your camera, just twitching every now and then. Yeah. So in my PhD in this presentation, I'm going to look at kind of like break it up into 3 different parts. So the first part is looking at what's driving the distribution of those 4 mammal species. So like site occurrence, you might call it, whether you saw the animal at a site or not. The second is looking at feral cat density. And the third is a spatiotemporal kind of activity index, which, you know, brings in some of the animal behavior, I guess. So yeah, I kind of like to think about this as like occupancy, density, space-time activity. So the first one, like there's been a lot of research in these environments. So we know things like rainfall, fire, different vegetation types, fragmentation, ruggedness all affect these mammals. And these monitoring programs, yeah, so I think that the difference in this study, was that I wanted to account for all of these things while testing the effect of fox control, and in particular, looking at like how many baits there are in the landscape rather than just fox control or not. And I'm not going to go into all of this stuff. I'm just going to pick out a few interesting things. So for foxes, I found that in the Glenelg region, which is in purple, where there was like in sites without fox control, there was foxes everywhere. About 70% of sites you'd go to, you'd see a fox. But as you start chucking out more poison baits in this landscape, we're seeing a near zero probability of detecting a fox at those sites. So it shows that like it's a really effective way to control foxes by, yeah, this poison baiting. And this is like, yeah, a really continuous bait replacement. In the Otways, there was less foxes to kind of start with, like there was less fox occupancy to start with. But yeah, we did see an effective bait suppression, which makes sense, like, but less of an effect, but which makes sense given foxes have only been controlled for like 1 to 3 years relative to Glenelg, which was much longer term. And in the Glenelg region, we saw a really positive response of long-nosed potoroos to the fox control. They really seem to be a really important factor for why potoroos are hanging on in this region. Didn't yet see that effect in the Otways, but still early days. However, we didn't see much of an effect in either region for southern brown bandicoots. They weren't really responding to fox control in this study. So maybe that's because there was more cats, but looking at cat occupancy, there was no effect of fox control on it. Cats were pretty much everywhere across these forests. I think like if you chuck out a camera for long enough, you'll see a cat there. So I really wanted to rather than look like, so maybe where cats are in the landscape, but everywhere there's not much, you know, how to, like, maybe occupancy isn't a great way to look for a positive response for cats to foxes, fox control. So the next, like a really big part of my PhD, and this next bit I'll talk about, is about cat density. So cats are really cool, feral cats, because they have these unique stripe and spot patterns on their coats. Well, most of them do. And we can use those patterns to identify individual cats and therefore estimate their density and track their movements throughout the bush. So I first grouped, I had about 25,000 images of feral cats, which I tried to identify the individuals in. I started off by breaking this up into like spotty cats and stripy cats and weird mottly cats and all that. And then, yeah, I spent much of my lockdown going really crazy looking at cat photos all day, every day. And so you can see here's an example of one cat that I saw at 6 different camera trap sites. Yeah, so that lets us estimate cat density, which is, yeah, often what we're interested in. And so this is just showing these sites in the, so this is kind of like, I stuck out hundreds of camera traps in each of these red little polygons to complement the broad scale monitoring that was already going on to try and estimate this cat density. And they were close enough together so cats couldn't move between these camera trap sites. So yeah, so I had about 200 camera traps at a time, and looking at one grid in sites with fox control. So these black dots are the poison baits. And so comparing sites with fox control to sites without, monitoring at the same time. So in the Glenelg region where there was long-term fox control, I replicated a control impact experiment 3 times. So I surveyed these two sites, took them out, surveyed these 2 sites, and with the help of DCART, we surveyed above and below the river above and below the lower Glenelg River. Yeah, so comparing what's happening to sites with fox control, without fox control, the caveat here is that we're assuming any differences in those sites to be from fox control. Whereas in the Otway ranges, we were able to get in before fox control started, and we monitored these 2 sites, and then twice afterwards annually. So this shows in the Glenelg region, a site without fox control compared to a site with fox control. And these different colours are the different cats that I identified, and lines show their movements throughout these large camera trapping grids, which are the red crosses. So I repeated this 3 times. This is the first time, 2 more sites, and 2 more sites as well. And so I found that in 2 out of 3 of these paired grids, cat density was higher in the sites with fox control. So these red dots here, this is cat density. But there was the second replicate, there was no difference in cat density between those 2 sites. This plot just shows the difference in cat density in the site with fox control relative to the paired site without fox control. And so this is about a 3.7-fold increase, difference in cats between these 2 sites. So yeah, it kind of, so I guess, yeah, it kind of begged the question of why, 2 out of 3 times wasn't consistent, but it looked like it was really mirroring what was going on in the foxes. So this is a plot of fox occurrence probability in each of these different sites. And you see that where we had the really strong effect is where foxes were really common in sites without fox control, but in the paired site without fox control, they were really heavily suppressed. So the biggest effect here. Whereas the site we didn't see any effect, which is Hotspur and Mount Clay, same kind of colours, there wasn't much of a change in the fox numbers, fox occurrence probability. I think that's just because Mount Clay was a really small fragmented forest block. It's hard to keep the foxes from coming in from the surrounding farmland. But yeah, so in the Otway ranges, this was before fox control started, our 2 sites. And then 6 months after fox control started. So you've seen a lot more cats here, and also monitored again a 1.5 years after fox control had started. So a pretty small time frame to monitor for changes in animal density. But yeah. So what we saw here is that in the sites with fox control, again, we've got cat density here and then the different years, and this is when fox control started. So before cat density, we're not assuming there's the same cat density in each of these sites. We see a bit of a difference, but in the site without fox control, cat density slightly declined over time, whereas in the site with fox control, it actually increased. So this plot, we're looking at the difference between those sites. In the sites with fox control relative to the site without fox control. It's fine to have a difference, but what we see is that the difference between those sites is changing over time. It is increasing. This is quite a small difference. It's quite a small difference, but by 2019, there was statistical evidence to show a difference, that the difference had changed. And again, another line of evidence for this was that actually foxes were again showing the opposite pattern. So in the site without fox control, they're actually increasing, and the site with fox control, fox occurrence probability declined over time. And again, another line of evidence. So this is cat density in each of the landscapes. So each landscapes, as well as the fox occurrence probability in each of these landscapes plotted against each other. So we see a negative correlation between cat density and fox occurrence probability across both those sites, actually quite similar. So normally, I guess this might just be because cats and foxes, one site might be good for cats but bad for foxes. They might different things, but there's a bit more evidence in this study because this scale, this gradient of foxes was artificial, like we manipulated it through the poison baiting. So it gives us And so you might realize that, you might notice that there's a lot more cats, and especially in those plots, it's quite obvious to see that there was a lot more cats in the Otways compared to the Glenelg region. And this was probably like a really, this is probably one of the key findings from my PhD, because previously in all the literature, it was expected that cats were basically absent from wet rainforest environments, but there hadn't been pretty much any studies previously of cats in these environments, which is probably why. So I wrote a short research paper about, maybe we might be overlooking the effect of cats in these really wet, dense rainforest environments. I also saw in the Otway Ranges there were some behavioural changes of cats to foxes, and it kind of looked like before there was a change in cat density, cats changed their behaviour. So cats kind of became less detectable as fox occurrence probability increased, and it also moved around a lot more as it increased. And there was kind of seemed to be this threshold of like halfway through the fox occurrence probability that's only when the density started decreasing. So it kind of got me thinking about this kind of relationship between behavioral changes and density. It's a bit of a stretch, but I was kind of thinking about that. And also looking at all these cat photos, I was also noticing some cats really just came back to the same site at really the exact same time, and went to different sites at different times. So I wanted to look at what cats were the dial activity of cats when they're active during the day, and whether they were avoiding foxes in space or time. So on average, they were pretty much doing the same thing. When we use all these camera traps and we draw some plots of when cats and foxes were active during the day. So this is sunrise and sunset, the middle of the day. Then we're both mostly nocturnal. But I did see that over time, there was a change in this behavior across space. And so I use this to break it up into, so this is the Otways as an example. And so I broke it up into the Glenelg region, which was dry vegetation types, and the dry and wet vegetation types of the Otways. So foxes were mostly nocturnal across the dry vegetation types, but kind of out all the time during the day in the wet forest. And I found that where fox activity was low, cats were mostly nocturnal, crepuscular, but as there were lots of foxes on those same camera traps, cats actually became more active during the day. but we didn't really see a change in the actual overall activity of cats, which kind of signals that cats might be avoiding foxes in time at those particular sites with high numbers of foxes. So in summary, I found some evidence that fox control might increase cat density and alter cat behavior. Like, I don't want to say tons of evidence. It's definitely not conclusive. There's a lot of assumptions in those experimental designs. But I do think that this research really shows the need that when we do fox control, we should be monitoring how cats are responding as well. And really, I think we often monitor the big prey species, which we know foxes love to take, but I think it's important that we also should monitor some of the smaller species, such as antechinus and rodents, which are more susceptible to cats following fox control. I think that's a pretty big missing kind of gap in this story. And more like technically, I think some of these broad species really missed these important interactions between cats and foxes, which might explain some of the why some of the previous research, kind of missed. It was a bit confusing. And last month, I published a paper in the Journal of Applied Ecology about this. So if you want more information, you can read that as well. But yeah, I might leave it there. Thanks.
Oh, Matt, thank you. More applause for more important and also sobering work again. Like, this isn't the most cheery ARI seminar session we've had, that's for sure. And I'm just realising that belatedly probably should have put up a trigger warning at the start for a lot of people seeing all those cat videos and cat images. Like, that's pretty awful. So apologies to everyone who was a little bit stressed out by all those cats. Okay, Tess. Questions for Matt. What have we got?
Yeah, so we've got a question here about how Gary's asking, how did you control for differences between the sites in terms of vegetation cover, proximity to roads and disturbances?
Yep. And I'll just quickly say to Fern, like, while, lots of cats ever in our bush, like, there is like the response of potoroos and stuff to the fox control. I think there is some really important positive stories from from the fox control. So it's not all bad. But yeah, it was quite concerning seeing the amount of cats. So the question was kind of referring to how did I control for different environments when I was looking at these paired landscapes. And so we designed the study landscapes to be as similar as possible while maintaining spatial distance, while being far enough that they were kind of like distinct. So kind of, walking that balance. So the Lower Glenelg River is a really good example of that, where it's mostly separated by the river, but it's kind of above and below. And in the models, I also looked at the effect of broad vegetation type on cat density as well, accounting for that. It wasn't a big driver in the cat density models, probably because these landscapes are quite small. All of these cameras were off the road, and that was really purposeful because we have good information that cats and foxes use roads to move around the landscape in. But I was really interested in what's like, they might be just using it to pass through, whereas they might not actually go off the road and prey on those native animals. They might So I wanted to have like a picture of what's going off road in the bush. But yeah, I think there's some research hinting that a response of cats to fox control might be that they might use the roads a bit more. So I think that would be interesting to look at as well.
Yeah, it's fascinating. Another question here is asking about whether there was any, or whether you have any knowledge of the relative impacts to wildlife in these different areas.
To the broader mammal, I guess the broader community. The cameras picked up a lot of different species. Probably one of the hardest things in my PhD, I got like 1.5 million camera trap images, and then I collated like way more data from both government agencies. And I found it so hard in my PhD to like boil this down into like a simple story. Like there's so many avenues and so much data. You can probably tell from that talk that went on for way too long. So yeah, there's a lot of species that we could analyze, and there's other species there we've got the data for. A lot of the data is being analyzed and has been analyzed. There is a bit more information there. Yeah.
I've also got one interesting question here about, is there any evidence of pig predation of other small marsupials? Could they potentially be another predator, I guess, is the question, in these landscapes?
Yep. I think they could be, yeah. So they were present in a couple of the landscapes in the Glenelg region. They're now kind of expanding in the Otway Ranges. So yeah, I think, like, I put up that picture in there of the pigs because it was really one of the things that, going out and doing this field work, seeing the damage that pigs were doing in these landscapes, it was really, for me, I see pigs as a massive priority now, which I kind of thought, didn't really think of as much. So I think that is a massive problem, yeah.
I think that might be, I think I've covered most. There's a couple of questions about 1080, but I think probably not your area of expertise, I'm guessing, Matt.
No, I was just researching the effects, the outcomes.
Yeah. Did you find rabbits? Sorry, the questions keep moving. And the movement of rabbits and foxes. What is movement with rabbits and foxes is a question.
Yeah. So in the there's really few rabbits in these studied landscapes. So in the wet forest, the Otways, I didn't see any rabbits. I didn't detect any rabbits. They're definitely present on like the farmland surrounding it, but they're not really in there. And same in the Glenelg region, only like there was quite low rabbit numbers, maybe very few of the camera trap sites, very, very few actually had detected rabbits. And the ones that did were really close to the farmland. So rabbits weren't, I would say that rabbits are not an important food source for cats and foxes in these environments, which is very different to the rest of Australia. And there's some really interesting, good research that shows how rabbits affect the interactions between these two species that, exists.
Alrighty, I think that we might leave that there for the questions, and I'll pass it to you, Fen, to close up.
Thanks so much, Tess. And Matt, I just did a quick look for your mesopredator paper. I've typed one in the chat. I don't know if it's the right one. If there's others that you want to share with people, please go and pop them in as well. Thank you so much. I mean, there are a lot of questions all around, well, both these talks, but just thinking about Matt's just now, like, there's an expectation that you can answer all of the tangled connections that are part of this, and there's so much that we need to continue to explore and understand in this for the ultimate aim of looking after wildlife out on country and in the bush. So look, thank you for sharing with us about your work today. Thank you so much to Graeme as well. Thank you to everybody who typed questions in the chat, to all of the people who came along and listened in. Tess will share this as a recording within, I don't know, a week or so, something, Tess. I don't know how long that process takes, but she's super efficient at it. So look out for your emails, and if you miss that, just jump on the ARI website and you'll find it there with all the other seminars. The recordings are there. Encourage others, can share that around with others who might have missed it today, or you can go back and have another look, or encourage others to come along to our seminars next time. Thanks again to everybody. Looking forward to seeing you next time. Maybe we'll look for something more optimistic next time. I'm feeling quite deflated from this one, but really important work, and thank you so much for sharing it with us today. Thanks so much, everyone. Bye.